This conference is being recorded.
Hi, everyone. Let's start. Welcome to the holiday edition of VanBUG 2019. Today, I am pleased to introduce to you all Dr. Quaid Morris. Quaid is a professor at the University of Toronto at the Donnelly Centre for Cellular and Biomolecular Research, where he has an appointment in both molecular genetics and computer science. Quaid got his start in academia back at the University of Toronto, where he graduated with honors in biology and computer science, eventually moving on to a PhD in brain and cognitive sciences at MIT. Quaid is a CFAR AI chair, as well as faculty at the Vector Institute of AI in Toronto. And also, he is a associate investigator at the Cancer Research Institute in Ontario. Quaid's group focuses mainly on the application of machine learning techniques to an array of biomedical problems, including cancer evolution, post-transcriptional regulation, and also gene function prediction. Today, Quaid's talk is entitled Reconstructing Cancer Evolution. Everyone, please give a warm VanBUG welcome to Dr. Quaid Morris.
Oh, like there I am. I was like wondering how much I can move around, and now I know. All right. Do we have a pointer?
You have to use your mouse.
Oh.
Sorry.
Okay. Hi, everyone. So thank you. That was a great introduction. This is actually my second time at VanBUGs. Can't remember what I talked about last time. It was a while ago. But I'm excited to be back. And I'm going to talk to you about some work that my lab has done on analyzing cancer data from bulk tumor samples with whole genome sequencing. And it's exciting to be here because, the BC Cancer Agency is really kind of one of the main focuses for this type of work and in Canada. And certainly a lot of the cancer evolution work has come out of either UBC or SFU. And so I'd like to begin my presentations with a pop quiz, seeing as this is also part of the course. And so...
It's your kid. We're having some technical difficulties. Sorry.
Oh, does this thing suck?
Yeah.
Oh, sorry.
No problem.
Okay, so let's see if I have another one. The question is, you got a little bit of time to think about it, how many somatic evolutions are in your body? Everybody know what somatic evolution is, mutation is? Somatic mutation is something that you weren't born with, but your cells have acquired by dividing and or being subjected to some sort of mutational stress like UV light or smoking or eating or basically just living. Any guesses? So let me give you, anybody know how many copies of your genome are in your body? Let's take that one first. Is there any way, I'm moving away from this, I'm hoping there's a way to actually show my slides. Is there?
You could send me those, we're trying to find another download now.
Oh, okay, great, all right. So let's start with that. Anyone take a guess? How many copies of your genome are in your body right now? 10 to the power of 6. 10 to the power of 12. 13. Correct. 10 to the power of 13. Anyone tell me what that number is? 10 trillion. You have 10 trillion cells in your body that have a bit of your DNA. You also have like 25 trillion red blood cells. That's kind of an amazing number. Okay, so you have 10 trillion copies of your DNA in your body. All right. How many mutations do you think per copy of your DNA? When you copy a genome, how many mutations do you think you pick up? 100. That's actually a pretty good number. Yeah, it's higher than most people would guess, but let's say you do 100. So how many mutations does that mean? 100 times 10 trillion. People know what that number is called? That number is a quadrillion. It's 10 to the 16, okay? All right. How many possible mutations are there on your DNA? How long is your genome? Haploid. 3 billion bases. And so every base can be mutated in one of three ways. So there's 10 billion possible mutations. So there's 10 to the 16 mutations in your body, and there are 10 to the 10 possible mutations. What does that mean? That means, on average, you have at least 100,000 independent copies of each possible mutation in some cell somewhere in your body, right? So if there's like one mutation that causes cancer, you should have 10,000 different cancers. Sorry, I got that number wrong, 100,000 different cancers, right? That's a lot of mutations. Just for fun, how much DNA do you think is in this room right now? Like if you took the DNA and put it end to end, this is another great number. How much DNA do you think is in this room? Like just counting the human DNA, not all the like other stuff that might be floating in the air, going into your lungs right now. Nobody else thinks about that stuff? Maybe I think about it too much. Okay. The unit to express that number in is light years. How many light years of DNA do you think are in this room? Human DNA. And it's about 1/10. In our body, we have 1/100th light year of DNA. Right? So if you get a room full of 100 people, and there aren't 100 people here, there's more like 30, I would guess. So if you get a room full of 100 people, you have a light year of DNA in that room. It's pretty cool, right? Right. Okay. So how many somatic mutations are in your body? And we just went through that. There's more than 1,000 trillion mutations in your body, somewhere between 50 and 50,000 somatic mutations in the somatic cell. And we know this because everybody's born with 50 new mutations. And your germline is probably the most conserved part of your body, right? That's where all the energy for like repairing and preventing mutation is probably going on. There's parts of your body that your body just does not care about. Nobody cares about your skin cells. Your skin cells have 50,000 mutations because you go outside into the sun, right? And UV causes a lot of mutations. Okay, and there's a lot of evidence to support this number, and I put some of that evidence up there. And then the implication is that every possible mutation will, on average, happen more than 100,000 times somewhere in your body. And here I like to say, so if you care about how the human genome evolves, you should study somatic evolution, so evolution within your body, rather than looking at germlines, looking at germline mutations, right? Because if someone's born, every person has been born with like 50 mutations, there's been like 10 billion people ever in the world, so there's like more mutations in my hand and maybe more in my little finger than have ever happened in the human germline in our entire human history, right? And so that's something to think about, right? And so we think about that, and we got very interested in this specific problem. And the other interesting thing about the cancer in this context, because this is like somatic evolution gone wrong, but certainly that's where you get the most mutations, is the cancer genome is its historical record. Oh, there's my mouse. Okay, and what do I mean by that? Well, what I mean by that is because this is like mitotic cell division. Every daughter cell gets all the mutations that the mother cell has. So your cells are just accumulating mutations from when you were an embryo to when that embryo grew out to be an adult human and got your 10 trillion cells, and then those cells started replacing one another until one of those cells started misbehaving and eventually became a tumor and grew out to become a tumor or cancer. Okay? And so when you sequence a tumor, you're seeing the entire mutational history, not only of the cells in the tumor, the most recent common ancestor of all those cells, because there's one cell that started the whole thing, but also all the mutations that happened before, right? And to a first approximation, all of the cells in that tumor have that set of mutations that was in that first cancer cell, right? And this is not new. People have realized this for a long time, and people have spent a lot of time, decades of time, trying to identify the driver mutations. So what are the driver mutations? So these are mutations that give a cell a selection advantage. And when the cell has some sort of selection advantage, that cell grows and makes more of itself. And ultimately, when things go bad, that becomes a tumor and takes over your body. You know, the cancer is like, you can think of it as a single cell organism that's specifically designed to infect your body and kill you. Okay? They provide selection advantages, but because of the fact that they provide these selection advantages, they cause cells with those driver mutations to increase in frequency, to define what are called clonal populations, right? And that clonal population has not only the driver mutation, but has all the other somatic mutations that were in the genome when that cell acquired that clonal mutation. So they increase in frequency as well. They what's called hitchhiking. And these other mutations that don't provide these selection advantages are called passenger mutations. And so you can see them when you sequence the tumor because maybe they don't give a selection advantage, but they happen to be around when that cell gained a driver. Cells gain drivers, they increase in frequency. All the mutations that occurred before that driver also appear along with the driver. Cells gain another driver, some descendant of that cell increases in frequency. You get more of these mutations and so forth and so forth and so forth. On average, cancer has somewhere between 5 and 10 driver mutations or driver events, right? But when you sequence a tumor, and we've sequenced tumors people sequence tumors at like a reasonable depth, like let's say 60x, and do whole genome sequencing, you see on average 3,000 to 5,000 mutations, right? So what does that mean? So that means that every driver has hundreds to thousands of passenger mutations, right? So unlike the drivers, which are really rare, these are really common. And they're random. They're not really random. That's why I put them in quotation marks. They're random in the sense that they're not providing selection advantages. So you can say things about the cell state based on where they've accumulated. And I'll explain what that means in a moment. Okay. And so as someone with like a kind of more machine learning background, where we're interested in trying to detect weak signals, but we need a lot of data to do that, we decide to look at these passenger mutations, right? And try to understand what we could say about tumor development or cancer development by looking at patterns of passenger mutations. Okay, and so why do we think that that's possible? Okay, and so this is, what I'm showing here is something called the Vogelgram. This is one of these sort of very well worked out and now slightly controversial genetic models of the development of cancer, starting from like colonic stem cells that gain some driver mutation that then causes the cell that gained that to have a lot of progeny that then grow out to make this small adenoma. Further driver mutations cause the cell that gained those further driver mutations to grow out even more, making a large one. And ultimately, a few more driver mutations lead to this kind of carcinoma, tumor, frank carcinoma, what do you want to call it, right? So each of these driver mutations is gained in the background of a whole bunch of passenger mutations, right? And if you believe this genetic model, which is a stepwise change of phenotypic states, these passenger mutations are being gained in cells that have different phenotypes or are in different states or are different types of cells, right? And so the degree to which these passenger mutations are reflective of cell state, you might be able to reconstruct something about these intermediate states, right? Or maybe better understand the initiation and progression of cancer. And why is this important? It's important because driver mutations are really hard to find, right? There's a lot of different ways to break something. And so, you know, the Vogelgram here, this is something from a paper that I took from like 2013. So here, the original like Vogelstein model had specific mutations. And then it was to realize that you can't talk about specific mutations. These are mutations that affect pathways, right? And so now it's listed in terms of pathways. And, you know, to tell you the truth, really, for many cancers, we know maybe 1 or 2 of the 5 to 10 driver mutations. And after those 1 or 2 initiating mutations, things go a little bit awry, right? And there's a huge diversity of possible driver mutations, which are incredibly difficult to find because, you know, the best way of finding them right now is just to look for overrepresentation. And so you got to sequence a lot of cancers, right? But if what you think of is cancer as a sequence of phenotypic states, and the passenger mutations are being accumulated in those phenotypic states, maybe they might provide either a more reliable indicator of a specific phenotypic state, which would help you better find drivers, or maybe they can just replace this driver view of cancer itself. And so we decided to ask, well, to what degree are either of these two things true? Okay. And so to interpret these different cancer epochs that may or may not correspond to different phenotypic states, you have to group the mutations into these epochs, right? And so I'm going to jump forward a little bit and kind of give you an answer to a question that's going to come up later. Right. Usually I need water to oh, great. Hi, everyone. Okay. So the way I'm drawing out this tree shows that there is this successive gain of these driver mutations. And this last driver mutation is probably the one that was gained, let's say, in this cartoon image of cancer evolution by the most recent common ancestor of all the cells that are now in this tumor. Okay? Mutation doesn't stop when the tumor grows, and you get additional mutations. And these mutations are not present in all the cells, and that's why there's a little bit of a branching there, right? These are called subclonal mutations for reasons that are a bit complicated to explain. And then these are called clonal mutations because, to a first approximation, they're present in all the cells. Okay? Defining clonal mutations with these epochs is somewhat difficult because this cell has all of these mutations, right? But defining these epochs is easy because cells have subsets of mutations. So you can say, okay, well, these subsets are in cell A and these subsets are in cell B, right? So you can ask questions about different subsets by looking at subclonal mutations. It's harder to say things about clonal mutations, okay? All right, so what I talk about today, and I've set up this talk so I have 3 things to talk about, and I never get through 3 things, so based on how the audience seems interested in things, I talk about 2 of them. I'm going to start talking about defining epochs to the point at which I get a little bit bored of that topic. I'm definitely going to talk about inferring historical states of cancer, and I might be able to get to understanding pre-cancer evolution. And so that's like looking into these clonal states to sort of ask what the sequence of events are before we get to the point at which there's the most recent common ancestor. Okay? Great. I am happy to stop to answer questions or to clarify things that are unclear. I like an interaction during my talks. Okay. All right. So a lot of our work is pre-assuming that we're doing sequencing under a very specific approach. And so that's it's called bulk short-read sequencing. So we're taking a heterogeneous sample of the tumor, and there could be 1 or more samples, and these samples could be longitudinal over time, or they could be spatial, cut up your large tumor into small bits, and sequencing like this population of hundreds of thousands or millions of cells at the same time. So you're looking at a lot of genomes, and when you do short-read sequencing, even though we have a lot of mutations, mutation in a specific genome is rare, right? So if you have 3,000 mutations in a 3 billion base genome, you have 1 mutation per megabase. And so if you're doing short-read sequencing and you're sequencing a population, you're very rarely going to get a read that covers 2 mutations. So you don't have any linkage between mutations. All you get when you sequence this population is the count of how often or what proportion of the genomes in that sample have that mutation. That's called its varying allele frequency. And now that's a little bit difficult to work with, and so that's why we need special algorithms to try to recreate and build out this, what we're going to call this clone tree, which is these relationships between the different epochs of the cancer evolution. Got it? Okay. And I've been focusing largely on what are called point mutations, and so these are signal nucleotide variants. So these are substitutions, or these are like small indels. And then when you sequence populations, you can get what's called the allele frequency of the variant by just mapping reads back to a reference genome and counting how often that read has the mutation. But when you do whole genome sequencing, and to a certain extent when you also just do something where you select out the exons and do what's called whole exome sequencing, you can also get information about copy number aberration. So one thing that happens a lot in cancer is you get what's called aneuploidy. You get loss of chromosomes, you get gain of chromosomes. In about 1/3 of cancers, you get duplication of the chromosome, a whole genome duplication that then leads to a whole bunch of different loss. And so you can infer that information by looking at whole genome sequencing. You can also get a little bit more information about these gains and losses by looking at what are called structural variants. And that's where pieces of the genome that are supposed to be together are no longer together. And you have reads that are covering those areas, and that can give you information about how the genome was rearranged. So one thing that's very common in some cancers is you get a chromosome that explodes in something called chromosurpsis, and it gets knit back together in various different weird ways. Okay. We're going to be talking primarily about single nucleotide variants, but I just want to let you know that these things are present. Okay. All right, I am already bored of this. So we're not going to do this stuff. And you're going to hear about the last two topics, all right? Okay, inferring cell states from patterns and mutations. So based on what I've already told you about, I've convinced you that you can take data from whole genome sequencing and group mutations into different epochs. And now what we're going to do is we're going to try to understand what's going on in those specific epochs by looking at patterns of mutations. Okay, and so what does that mean exactly? Okay, so who here has seen a mutational spectra before? All right, less than 10%. Okay, this is really cool. Okay, and so this will kind of blow your mind a little bit. So you can take mutations that you see in a genome, which you detect through whole genome sequencing, and you can classify them into 1 in 96 different types. And those types indicate not only the type of substitution, and so we're talking about substitutions here, so C to the other 3 possibilities, or T to the other 3 possibilities. So there's 6 different possibilities because you can't necessarily tell what strand a mutation occurred on. And then whether or not a mutation occurs at a specific location in the genome often depends on what the 5' and 3' bases are, right? And so if you take those 6 possible mutation classes and multiply by 4, and 4 you get 96, and then you can just make a histogram where you count the number of mutations in each type. And as I told you, when you do whole genome sequencing, you gain between 3,000 and 5,000 mutations. So these histograms are called mutational spectra, and along the x-axis here we have the 96 different types, which you obviously can't read, but you can see that the different colors correspond to different substitution types. And so the 16 bars here correspond to the 16 different contexts, and they're always in the same order. Okay? And so you can look at, so looking at the spectra, I can say a couple things immediately about the cancer or the person that the cancer was taken from. So this is a mutational spectra of a breast tumor. And I can tell you it's probably taken from an older woman, right? This is a mutational spectra of a lung tumor. I can tell you this is probably taken from a smoker, right? I know that this is taken from an older woman because these 4 bars, this is a bit like palmistry, are a little bit higher than you would normally expect. And these mutations are CG to TG mutations. It's a very common mutation that happens at a methylated CPG, right? It's so common that the human genome is depleted for CGs. And wherever you see a long track of CGs in the human genome, that's usually an indication that that region is important for, often it's a developmental promoter. It's called a CPG island, okay? And that's because these are very classic mutations that just happen at CGs almost naturally, methylated CG regions. But they accumulate at a rather constant rate as people age. And so there was an earlier study that showed that they're very highly, the number of these types of mutations is very highly correlated with the age of the person that the cancer is taken from. So we have a clock mutation. These mutations down here, C to A mutations, are caused by tobacco mutagens, right? And so if there is, or smoking mutagens, probably not tobacco mutagens, but the processing thereof, the tobacco. And so if you see a lot of mutations, that's indication of a smoker. And the reason that we know these specific things, and these are mutations that are associated with the NAPO process. The reason that, and so if you look at tens of thousands of different tumors, and you look for patterns of mutation co-occurrence, as was done by Ludmil Alexandrov when he was in Peter Campbell and Mike Stratton's lab, you can identify these, what they call these mutational signatures. And they took these to represent different mutational processes, right? And so these are the timing mutations, the CPG mutations that I was telling you about. These are these like 4 special bars. Over here are the mutations that are associated with smoking. And these mutations, when they looked at them, were present largely in lung cancers of people who smoke, but like other internal organs that might be exposed to tobacco mutagens. These mutations here, this so-called signature 7, these are mutations that are caused by UV light, and this group of mutations is only seen in melanomas. Okay? And they defined 30 of these so-called mutational signatures, which were these patterns of co-occurrence, surprising patterns of co-occurrence of mutations. And this was the first initial paper 6 years ago. Now, 6 years later, there's probably about 100 different signatures. There's about 50 substitution signatures. I think there's a few dozen indel signatures. There's also signatures of types of structural variations that occur. And so these different signatures represent either mutations that are caused by specific cellular stresses, or either these are external mutagens, but many of them correspond to internal mutation repair processes that are being stressed in some way, or either because they are defective, because you've gained a mutation that injures that pathway, or because the cell is dividing faster than it normally would. Okay. Yeah? Right, so UV light causes like a very specific mutation. It causes a CC to TT mutation. Exactly. So these are and so I should say that these signatures were defined computationally and then interpreted based on these processes. So these signatures were defined by taking a large matrix, which contained all these counts, these histograms, and running something called non-negative matrix factorization, which would basically identifies co-occurring groups of mutations across the multiple samples, right? And so this is one co-occurring group of mutations that just happened to show up primarily in melanomas that was immediately interpreted as being caused by UV light because it was consistent with what was known about the mutational processes caused by UV light. Okay? Yeah.
Take one profile and then pull it.
That's a great question, and I'm trying to, I don't know if it's the next slide or like 2 slides from now. Okay, it's the next slide. All right. So yes, they are additive. So there's multiple mutational processes, and remember, the mutations that you see in a cancer, they're a summary of the entire history, right? And so there could be a bit of UV exposure that could then lead to some defective mutation process or some cellular response to the damage that you see, right? And so you would expect mutations due to multiple processes. And when they did this non-negative matrix factorization, that technique is a bit like principal component analysis, and it allows you to detect additive contributions of different sets of mutations that are associated with presumably different mutational processes. And so you can estimate what are so-called signature exposures by taking this mutational spectrum and doing like basically constrained linear regression to estimate the proportion of mutations in that spectra due to each one of the signatures. So it assumes like strong additivity. They're exactly additive, and that's what people think. I mean, it's because these mutations are kind of occurring independently over a long period of time. That's what makes it additive rather than like nonlinear. Okay. Right. All right. So we're going to jump right into it. So as I pointed out, the tumor is a historical record, and based on the information I told you in the part that I skipped, you would immediately realize that mutations that are more common in the population that have higher variant allele frequency are the ones that occurred earlier, generally speaking, and the ones with lower variant allele frequency are the ones that occurred later, so that you can come up with this sort of a rough timeline of the order in which mutations were occurred by just taking their allele frequency in the population, potentially correcting it for copy number changes that will change the relationship between cellular frequency and variant allele frequency. I'm just saying that in case there's people here who know what I'm talking about. and then order the mutations roughly by time. And so what Yulia Rubinova, who's a student in my lab, did is he said, okay, well, let's look at the change in exposures over time. We have thousands of mutations. We can estimate the exposures at different time points. Let's look at what happens when you do that. And so she developed an algorithm that we call TrackSig, and that's essentially what it does. And so what each one of these lines here is the exposure, or the proportion of mutants contributed by a different signature at a different measure of time. This is like the x-axis here, the label of the x-axis is average number of mutant alleles per cancer cell. But if you have more copies, if the population has more of a mutation, that mutation generally occurred earlier. And so we're using that as a sort of a surrogate for time. So these are the earliest mutations, and these are the latest mutations. And the signature exposure is what proportion of the mutations on the y-axis are due to that. And each one of these lines corresponds to the exposure trajectory of a different signature. And so the signature 9 is, these are mutations associated with it's like a hypermutation, somatic hypermutation process that occurs during B cell development. That's due to like basically, yeah, I'm not even going to say it. Okay, and so this is an indication that the earliest cells were ones that were undergoing this type of maturation. But as the cancer, this is a type of leukemia, developed, increasingly a larger fraction of the mutation came from a different signature, this like signature 5, which is this kind of unknown signature of unknown etiology. People don't really understand what it is. And then there's another signature here. So we have these. So we were able to establish the trajectories over time. And so what Ian Shih, who used to be an undergraduate in the lab and now is an undergraduate, did is he identified like an optimal, he found this optimal segmentation algorithm that we're using for everything. And its name is PELT, Pruned Exact Linear Time Segmentation. And we love this algorithm because if you have a 1D signal and you want to do segmentation, it runs somewhere between quadratic and linear time, and it's an extremely useful and very flexible algorithm that you can employ. And so we segment everything. And so here what we're doing is we're segmenting these exposure trajectories into ones that have constant time. And that allowed us to detect these boundaries, or so-called change points, where the signature exposure changed. Right? And so when we looked up these boundaries, or these change points, oh, okay. Yeah, we're going to talk about something else first. Okay, great. And so we did this analysis within the context of a larger project called the Pan-Cancer Analysis of a Whole Genome Project. And so this was a project that was joint between researchers in the ICGC, the International Cancer Genomes Consortium, and the TCGA, which is the American version of the ICGC. So it's the world and America. We collectively, we being 800 researchers, analyzed 3,000 whole genome sequences. And we were members of a group called the Heterogeneity and Evolution Group. And us and one of our colleagues, we collectively analyzed the timing of certain events within tumors. And so because we had 3,000 whole genome sequences from about 30 different tumor types, we were able to identify patterns that were consistent among multiple tumors of the same type. Did you have a question, or you like just putting your hand like that? Okay. All right. Okay. And so one of our colleagues developed this like extremely complicated but useful once you look at it visualization. And so what this is a visualization of the change in signature exposure of tumors of a given type over time. And on the left side are the signatures that were high early on and decreased, and these ones were low early on and increased. So this says, and the height of the bar here is how much they decreased, and the opacity is what proportion of the signatures in the group decreased. Okay? And so what this says is signature 9 decreases a lot in most tumors of this type, or most cancers of this type, Signature 5 increases a lot in most cancers of this type. Signature 5 decreases a bit in some of the cancers, and signature 8 increases a bit in many of the cancers. Okay? I want you to look at this and realize that you can identify sort of the overall pattern of signature change by just visually looking at how these things look, because now I'm going to show them to you for like 30 different cancers. And what you can see, go. Oh, blood reference. Yeah, there's all blood reference. Yeah, yeah. So these are not. There's not. It's relative relative to the germline of the person. So they collected both blood. They did sequencing at least 30x, I think, in the blood, and the tumor sequencing was somewhere between 30 and 50x on average. Okay, so these are all somatic mutations. Yeah. It'd be a lot less interesting if you looked at like germline mutations. There's not as many of them, and the signature exposure are a little bit different. Yeah. Do you look at germline mutations? Oh, okay, okay, great. Let's talk about it later. All right. So what I want to show you and convince you of is that every one of these tumor types has a distinct pattern of change of signature exposure. Right? Not only these distinct signatures, but there's a distinct pattern of change. So in ovarian cancer, there's a signature called signature 3 that generally goes up, and that's because signature 3 is associated with BRCA1 mutations, which tend to be somatic in ovarian cancer and tend to be germline in breast cancer. Right? And so this signature trajectory, in some ways, it's a representation of cell state. Right? It's telling you something about the external mutational stresses. It's telling you something about the internal mutational processes. You can sometimes deconvolve out information about even age of the tumor and the age when certain events occurred, just by looking at these patterns of types of mutations and how those patterns change over time. Okay, so that's one way in which somatic mutations are telling us something about cell state. Okay, great. Questions? All right. Here's another way that somatic mutations are telling us something about cell state. Okay, who's seen this picture before? Okay, this is a pretty awesome picture too. All right, this is a picture it's a figure taken from a paper from Shamil Sunayev's lab. He's at Harvard Medical School, and the first author is Paz Polak, who is now has his own independent position in Mount Sinai, New York. Okay, and so what is this a picture of? So along the x-axis is spatial location in chromosome 2. So you can see chromosome 2 up here at the top. Okay, and there's two lines here. So the black line is mutation density in a set of a few dozen melanomas. Okay, so this is how often you see a C to T mutation at that spatial location. The blue line is something called chromatin inaccessibility in melanocytes, which is the presumed cell type, was the cell type of origin for a lot of melanomas. Okay, and so where things are high in the blue, that means the chromatin is more closed, means it's more inaccessible. It means it's wrapped up and is not being actively transcribed, is one way to think about it. And so you can measure this accessibility using an assay called DNase-1, hyperaccessibility, that cuts DNA that it can access. Okay, the blue line and the black line are basically on top of each other, right? And so they're very highly correlated with one another. And so what does this say? Well, it says that mutations accumulate in areas of closed chromatin. And chromatin is a representation to a more or less extent of which genes are being active and which genes aren't active. It's a bit more complicated than that. And then in this paper, they wanted to show on that there were associations between chromatin marks and mutation densities. Since this work has come out, there's been a lot more elucidation of that association, right? And so mutation rate depends on chromatin state. And chromatin state changes between different cell types, right? So if you look where in the genome a mutation occurs, that could potentially tell you something about the cell, the state of the cell that the mutation was gained in. Okay. So a student in my lab, Gurneet Atwal, tried to use this information as a way of, well, we first tried to use this information as a way to make inferences about chromatin state and cell state, and we realized that it was too hard. And so we asked, is there any information in here at all? Because in this previous work, they were looking across dozens of tumors, and we want to say things about one single tumor, right? And to say something about one single tumor, Gurneet started to recreate what they had originally done in Shamil Sunayev's lab's paper, where they split the chromosome into 1 megabase regions, and then they counted the number of mutations in each one of those megabase regions. And that makes a vector that's got 3,000 different entries in it that contains these counts, okay? And then he made another vector that contained these SNV types. And he said, based on these 2 vectors, can I recover what tumor type this is coming from? And he did that as a way of saying, well, how much information is there actually about cell state for individual tumors? Is there enough that we can recover tumor type? And why were we not sure that this would happen? Because individual tumors, this is a vector that's got 3,000 entries in it. I told you there's like 3,000 mutations on average, so this vector is going to be very sparse. There's going to be a lot of zeros in it. Most of these entries are going to be 1, right? Can we actually say anything at all? And so while Gurneet was doing this, we found out one of our colleagues was also doing exactly the same thing in collaboration with Paz Polak, and his name was Lincoln Stein, and he's at the OICR. And so we joined forces, right? Now, before we joined forces, Wei Zhao, who's an analyst in Lincoln's lab, had tried to use these vectors to predict tumor types. So we had 24 different tumor types. We had like thousands of these vectors for each of these different, well, across the whole set, maybe hundreds or dozens for each of the tumor types. And he trained a random forest, which is a classifier, to try to predict tumor type. Now, he was asking a different question. He was asking not only about the question about how much information are in these somatic passenger mutations, but to what degree can you predict tumor type from drivers? Okay, and so what Wei found, and so this is the performance of the different classifiers that Wei trained. Each one of these points corresponds to a different cancer type. There's 24. And these are looking at different types of features. And what's on the y-axis here is something called F1 score, which is kind of like the measure of an accuracy of a classifier. Right? It combines recall and precision. Higher is better. And you can say, when you took a representation of all the driver mutations that have been detected in this tumor type, and there are 500 different driver events that were defined by our colleagues in the PCOG, he saw that there was very less classification signal. So the accuracy was somewhere around 25, which is still pretty good because there's 24 different tumor types. So like average accuracy would be somewhere around 5. But the accuracy was much higher looking at the mutational spectra or the SNP distribution. And what we were doing in collaboration with Wei was training a deep neural network. You're looking at both the distribution and type things. We could get a performance that was much, much higher, right, by combining those 2 things together. So we're looking at a performance somewhere around 80 or 90%. It's going to be a bit clearer there. When we added the driver mutations, performance really didn't change. So this is the median. Median performance went up like maybe a tiny bit. It wasn't statistically significant. The average performance went down. So we interpret this as saying like either we were overfitting a little bit or there's just no additional information in these drivers that's not already captured by these mutational distributions and this spectrum. Right? And so this is what's called the confusion matrix. And this says there's 24 different categories, and this is the predicted answer. And the number inside the matrix is what percentage of the kidney RCC tumors were actually predicted to be of that type. And so you see there's some tumor types we do very well on. There's some that we don't do as well on. But where the classifier makes errors, it's making errors often. Let's see here. This is a good one. For cell types for tumors with at least the same initial starting cell, so 2 B-cell tumors. Or on these tumor types, esophageal and stomach tumors, they're hard to distinguish, and there could even be clinical errors because there's a lot of tumors that kind of live in the boundary between those 2. Okay. Now, here's so the overall accuracy was like 91% over these 24 different types, and like up until that point, the previous accuracy had been reported for both DNA-based classifiers And RNA-based classifiers, this is RNA extracted from the tumors that you would think would be a bit more predictive, this was competitive, if not better, than all of them. But the nice thing about the DNA-based classifier is that when tumors metastasize, they keep all the mutations with them, right? And so we took the same classifier and just at the time in which we put this into archive, a second paper came out in archive from the Hartwig Foundation where they had sequenced about 2,000, in whole genome sequencing, about 2,000 metastatic tumors that were actually post-treatment. And we could immediately apply just the same classifier out of the box to those tumors, not being trained on those. And we got an overall accuracy, and this accuracy is actually wrong, and I should change this. It's about 82%, right? And a lot of the differences came from the fact that we think that the mutations were processed in a slightly different way. And then here's the confusion matrix, yeah. Yeah, so some of these cancers are from the same tissue. So the B-cell tumors are from the same tissue. It does have some trouble distinguishing, but it's not impossible. Some of the CNS cancers are from the same tissue. Yeah, there's a couple of them that are from the same tissue. We can make distinctions between the same tissues. So it's a great question because like about half the mutations are accumulated before transformation. Right, so many of the mutations are indicators of tumor type, but there's at least some mutations that, indicators of cell type of origin, but at least some mutations that distinguish between tumors of different types of the same type. And so we've applied this to like breast subtypes, and I think we do well. Okay, all right. I was only able to tell you about one thing. So is there any questions about that? Yeah. Yeah, I mean, I think the same factors that cause passive mutations cause driver mutations. But that's what I'm arguing. What I'm arguing is that different cancers are subject to different mutational stresses, and the types of mutations that you see are readouts of that. And so not only do the types of mutations that you see depend upon what the mutational stresses are, right? Whether they're exogenous, whether they're endogenous, whether they're like failure. Most of the mutations that you get in your cells are not copying errors. They're like failure to repair DNA damage, right? And so if DNA damage repair is not working, or if it's stressed in some way, you're going to see accumulation of the types of mutations that DNA damage repair process would normally think. So that's telling you something. What it's telling you we're not entirely sure about yet. But also where mutations accumulate depend on cell state in a way that there's a relationship with chromatin. Some mutations are accumulating more on antisense versus sense strands because the process that repairs those mutations is transcriptionally coupled, right? And so all these types of information that are telling you various, maybe indirect and hard to interpret things about cell state are contributing to what somatic mutations you see and where you see those mutations. And then further, if we can add the information about time, we can start to go back and look at the historical record and ask questions about those stresses over time. Yeah. We're not sure yet how much information we'll get out. And so what I've been trying to tell you is there's some information there, and it's to some certain degree predictive, and it's more predictive than we would normally expect it to be. Okay, and so I'm not going to tell you about this. I just copied these slides from Kim, and you can tell Kim, who's this amazing student, as co-supervised with Philip Awadawa, who's looking at clonal hematopoiesis, also makes great slides with a lot of animation in them. Okay. All right. So what I told you about today was how to define epochs to do subclonal reconstruction, and you're all convinced about that. And I spent a lot of time talking about inferring historical cellular states from patterns of mutations, which is something I'm super excited about right now. And this is an open field. This is a very exciting area. And this, place like BC Cancer Agency, which hopefully is continuing to generate a lot of whole genome sequencing. There's a lot of data here to analyze, right? And, you know, it's the passenger mutations, for me at least, are the most interesting ones. Yeah.
So if we go back to chromosome 2,
yeah.
chromosome 2, and there's a correlation between passenger mutations
Chromatin.
and the chromosome mutation.
Yeah.
So now, where I understand it, that like each cell, different cells in the body, different expression part of that genome is expressed.
Right.
So if a mutation happens to hit a location that is not expressed, hit a location that is expressed, it might cause cell to die, or it might not. So this might lead to a different.
Well, the cell repairs mutations all the time. There's like tons of cellular damage. There's tons of damage to DNA that's constantly being repaired. So mutation, you hit a transcribed region, isn't going to kill the cell. I mean, if it's a mutation that like immediately causes cell death, sure. But like, you know, it's damaged. Yeah. Yeah. You know, it's I mean, as I presented in the third part of my talk, there's like, you know, most of the mutations are neutral, and the other ones are like maybe slightly damaging. And one of the things that distinguishes people who are in precancerous states from people who are healthy is not the presence of driver mutations, but the absence in those cells that have the driver mutations of damaging mutations, right? But like, just because you have a mutation in a transcribed region is not going to kill the cell under almost all circumstances. It's very rare that a single mutation is going to do something about, I mean, you also have 2 copies of every gene as well, right? Yeah. Yeah, I mean, there's been recent work from Steve Jones's lab that built a classifier, and then there's also work from OICR that builds classifiers. RNA-based classifiers work pretty well, and probably they might work a little bit better than what we presented here. But the thing about RNA is it's a lot harder to get than DNA, right? And there's good reason to believe that the RNA expression levels are going to change a little bit when the cellular environment changes, whereas the mutations are staying pretty much the same. And so that's you know, where we think that the classifier of this type would be useful. Yeah. Yeah. Oh, it's how the mutation is being called. Okay. Right. And so like there's a lot of variability in like what mutations get called by various different pipelines. And so we didn't look into this deeply. So we, you know, what we saw when we delved into what the differences were was that the mutational spectra looked very different for tumors from this metastatic cohort versus the primary cohort. And there could be 2 causes of that. One is treatment, and the other one is that they weren't using the same pipeline to call mutations. And that pipeline can change the mutations that get called quite a bit. Yeah. Possibly, yeah. I mean, the reason we think it's a pipeline difference rather than the treatment difference is that we also did a validation in a different set of primaries, and we got about the same performance as we did in the Mets. But yeah, certainly if you sequence deeper, you would do better. Yeah, we're happy to get whole genome sequencing, period. If you want to give us whole genome sequencing like 200x, I would be overjoyed. What? But you do get reversal in phenotypes. Right. Right.
How do you sort of see that? Well, I'm just trying to like wrap my head around it because we expect mutations to go one way.
Right.
And then if you get these, could you somehow all that information?
Well, maybe. I mean, so we're not super optimistic about being able to say things about specific tumors because there's just not a lot of signal. But we do hope that we might be able to say things about groups of tumors or do subtype things based on those groups. And then maybe by identifying things about groups of tumors, we can then go back and, subtype further or say something more about development that we can then, that gives us a signal that we could maybe detect the presence of differentiation. Yeah. I think it's both. And from this initial work, Mike Stratton's group has followed up, and other people have, to provide causes for some of these signatures. And then there's a recent work that's just come out where they actually took cell lines and they subjected them to different mutational stresses, and then they did the same types of analysis to find some further information about what these groups of mutations might be. But yeah, some of them might just be clustering artifacts, certainly. Yeah. But they're confident enough in these signatures that they give them numbers, and then those numbers stay the same. So there's an initial set of numbers, and now they rederive the signatures, and they were able to number them still. So some of them are fairly consistent across multiple different cohorts of tumors. I'm kind of hoping people will actually. Signature 5 is like the everything out signature. So there's some sort of low level of consistent mutation that people don't really understand. And signature 5 is very hard to distinguish from signature 3, and it's very hard to distinguish from signature 8. And how they're expressed in different tissue types changes a fair bit. So I don't think the story is done. So I think something's going to end up in textbooks a few years from now, but I think it might be a little bit different. And I hope there's not a nomenclature board that says, okay, well, this is signature 3, but this is where we are right now. Yeah. Yeah.
You have a mutation that just happens to match the, is an actual mutation that happens to match the allele that is in the and you're not calling it.
So, I mean, when you call mutations, you're calling against the germline reference. I mean, ideally, though some people try to avoid doing this, you're calling against the reference of the patient itself. So you take blood, and you sequence DNA from blood, and you sequence DNA from the tumor. So you have the patient-specific reference. Whether or not the mutation callers filter out known polymorphisms, I think that varies a little bit between the mutation callers, but I don't know the details deeply. Yeah. But there's not that many known polymorphisms. I mean, the frequent ones, there's 3 million. So it's 1 every 1,000 bases or so. Oh yeah, it's no problem. Sorry, things might have gone a bit long.
Let me just turn the recording on.
Yeah.